Hey, and welcome back everybody to the Sanctuary. I'm Professor C, and we'll be going through our series in Anatomy and Physiology. Today, specifically, we'll be talking about chemical reactions. So, let's get into it. Okay, let's do a little bit of chemistry today. Specifically, let's do metabolism today. I know we've seen the word. Let's talk about it a little bit more in detail. If you have a string of objects linked together by a bond, or bonds in this case, we can illustrate that very simply like that. And we can say this long stringy thingy is a poly, many, mer thingies, right? We've mentioned this before. A polymer. And let's say that's something you ate for dinner last night, right? Or as any organism, what it might have eaten that particular day. So you consume some polymers, and then later inside of your cells, inside of your body, that polymer is broken down, right? And we call that type of metabolism catabolism, right? Remember we learned about how cats tear things up. So we can remember that catabolism, or catabolic reactions, take a polymer and break it into single units known as monomers. Okay, what we're going to see now is heat will come out of this, and that will be a minor point, but that's always going to be kind of like a tax that you would pay to the universe every time you want to do one of these catabolic or anabolic reactions, for that matter. You have to pay a little fee to the universe in order to get that chemistry to happen. So you lose some heat. More importantly to us, perhaps, is that when we break bonds, we're going to find out we liberate energy. And I'll just draw energy as a big circle E right now until we need to get more into it. But there's the catabolic process. We're breaking polymers into monomers and are retrieving a little bit of energy from that. And now if we liberate the energy here, the word is exergonic. Exergonic. Releasing energy is an exergonic reaction. It's another way of saying catabolic. We're also going to find, in order to make this happen, we need to add water to the mix. But that's not the end of the story. That's only kind of half of the circle. We can go on this side and make more of this idea. Why do we want, as an organism, to create monomers from polymers? What's the point of doing that? What's the point of taking a hamburger you ate last night and turning it into its individual components and breaking it apart through digestion? What's the point of all that? Well, the hope is that later, in another time, in another place inside of a cell, you can rebuild those monomers into, well, your own parts, your own body parts, your own cellular needs. And we'll talk about a lot of those structures in future chapters. But you have to make your body parts, your cellular parts, from something. They just don't come from nothing. And if you've heard the phrase, you are what you eat, it's completely true. So let's just show that happening. Now, we don't want to rebuild a hamburger we ate, so let's kind of change it up. We'll make a different little monster out of those monomers, but there's something I need for my cell. What is it? I don't know. We're just making it up. But the point is, I have a polymer that I've eaten and broken apart catabolically into monomers. This is an exergonic reaction. It released energy. Water was needed to power it. Guess what? On the other side, we need to put energy into this system. I know that because I know now that I'm making a bond, right? I know that costs energy. And here, of course, we break bond, if we want to be clear, and we're putting energy into this system. So this is called endergonic reaction. Endergonic reaction. One more thing we can do, if we put water in on the other side, we need to take water out on that side. And there you have a pretty good summary of metabolism. All right, let's start with those catabolic reactions, those reactions where we'll be breaking something apart. You could also call this a decomposition reaction. It's a synonym for a catabolic reaction, again, implying we're going to be breaking something apart, decomposition. Another thing you can call it, and this is where it does get a little confusing, is it's got multiple terms. You could call it a hydrolysis reaction. And again, we know the root hydro could mean hydrogen, but in this case it means water. And lysis is a great root word that you should put in your memory bank, meaning to break. So water breaking, we're going to use water to break a bond. So a catabolic reaction is a decomposition reaction, is a hydrolysis reaction. So I do see in my picture here, AB, that's going to be a polymer, and it's going to be broken into monomers. I see water coming in the system, and I'm going to throw some energy out of the system because I broke a bond. So let's check it out. Water is added. Yes, it is. We break apart bonds by adding water. This is called hydrolysis. Bonds are broken, right? You could say that's catabolism or decomposition. Polymers are split. Another way to say it, the longer chains are broken into single units called monomers. And the energy, circle E, is kicked out because we've broken a bond. This is an exergonic reaction, a release of energy. You can see it over here too. Here we have some sort of polymer with 4 monomers in the chain. And I'm adding water and I'm breaking the bond. This is a pretty good way of visualizing what's happening to that water molecule as it separates the monomers. So this is a catabolic decomposition hydrolysis reaction. The other thing that's kind of missing from this picture is we're going to kick energy out of the system. It would be an exergonic reaction. Okay, so this is the opposite of a catabolic reaction, is the buildup. We said a way to remember that is an anabolic steroid builds you up. So we must be creating something here. We're taking individual monomers, in this case A and B, whatever they might be, and chaining them up together into a polymer. I know it's not a very long one, but we're just pretending right now so we can get the hang of this. Water is leaving. This time, if we're going to create a bond, and we are creating a bond, water must leave and energy must come into the system. That was called an endergonic system. So let's look at our key terms. Monomers join to form polymers. Yes, it's an anabolic reaction. However, just like you can call a catabolic reaction decomposition, you could refer to an anabolic reaction as a synthesis reaction because we are synthesizing something. Bonds are formed. Energy is stored in the bonds. This is called an endergonic reaction. And water is removed through dehydration. Sometimes you'll see it combined and they call it dehydration synthesis reaction. It's the same thing as saying an anabolic reaction. In organic chemistry is what we're going to call it in a future lecture here, is taking these monomers and polymers and going back and forth, building them and breaking them and building them and breaking them across the course of a cell's life. Dehydration synthesis, I'm pulling out water, I'm forming a bond, I'm putting energy into the system, I'm going from monomers to polymers. All right, that's dehydration. You could also call this anabolic, right? Dehydration synthesis reaction is an anabolic type of reaction. Let's do the opposite one. If you add water to a system, you will break bonds. So in this case, we go from the polymer back to the original monomers. So we're breaking a bond by adding the water, hydrolysis. Energy is freed from the system. So that's an exergonic reaction. If you're talking about a reversible reaction, it just means that you can go back and forth, and usually you'll see that because the pair of arrows here will show you that. Whether you have water being added or water being taken away from the system, you can go back and forth between the monomers, and the polymers. This, of course, is how you would accomplish this in the body, is by adjusting the amount of water. All right, let's try to think about a reversible reaction. We saw one there at the end that was kind of non-specific. Let's make it more specific. Let's draw a polymer. Now, let's say it's starch, something we'll look at in an upcoming lecture. But everyone's heard of starch. It's a plant material. Let's break it up. And we'll find out that starch is made of units of glucose. So the polymer is called starch, and its monomers are called glucoses. This is our first example. So in the body, say you ate some starch and you wanted to break it apart into glucose. Well, we know what we have to do to break it apart. We have to do a catabolic reaction. We know we need to add water to the system. So it's going to be called hydrolysis. We could also call that a decomposition reaction because we're breaking bonds. Remember, we're breaking bonds, so we should see energy leaving the system. This would be an exergonic system. Again, if it sounds repetitive, it is. It's the best way to handle things like this, is just to say them to yourself over and over until they ring true. Now, what I'm going to do is the same story, but I can go backwards, right? I can go this way if I wanted to. I could do an anabolic reaction under the right conditions. Now, what I will have to do here is pull out water to make that happen. And that's called a dehydration reaction. And remember, I can add the word synthesis here too, because building reactions, anabolic reactions, are synthetic. So there's a dehydration synthesis reaction in which I'm pulling out water to create bonds. So I know if I'm creating bonds, I need to put energy into the system. This is an endergonic system. So there's an example of a reversible reaction using a specific example of starch going to glucose. Now, if I wanted to talk about a protein and amino acid, I could do the exact same thing, except just change. So watch me erase this. If I just erase the name starch and the word glucose, everything else will stay exactly the same. Let's add, for example, as a polymer, protein. Now, protein is not made of glucose. It's made of a different monomer called amino acids. If I wanted to break apart a protein, I would do it through hydrolysis. If I wanted to build a protein from amino acids, I would do it through dehydration synthesis reactions. So these are called reversible reactions, depending upon the amount of water in the system and the amount of polymers and monomers already in the system. These are reversible reactions. You can go both ways. All right. This one's called, in some texts, they call this a displacement reaction. And some they call it an exchange reaction. So what we're going to see here is bonds are going to be made and broken simultaneously. We're not going to go just from polymer to monomer. We're going to do some building and some breaking. So there's going to be some anabolic reactions happening at the same time as catabolic reactions. And phosphorylation is a really great example of exchange reaction. I know it looks very complicated here, but let's think for a minute. First of all, ignore this purple thing. And now we have some things. There's a red thing here called ADP. All right. And I'm going to put it in a little halo here so you see the whole thing. There's a red thing called ADP. Let's call him C, because we're going to do this reaction here, AB plus C. Let's just look at it. The green thing, right, the green thing, let's call him B, and we'll call this little yellow circle A. And so what we've got up here is this green thing with a P on it plus ADP. And that's our A, B plus C. Okay, so once you see it like that, it makes it a little bit easier to deal with. Now, what do we have on the other side? Well, B's left alone. Again, it's changed its name, but that's not the point here. It's still the green thing. It's still B, but now it's all by itself. It lost its P. So B's all by himself, but it looks like A and C have hooked up to become something new called ATP. Now we're going to discuss this further in another lecture, but that's the idea here. Now we have A and C. Okay, so now we have this green guy. I'm just going to call him Pep again, even though he changed his name, plus ATP. This example that I just shown you is called phosphorylation. It is a little bit beyond our pay grade at this moment, but we'll talk about it in other lectures quite a bit. It is the classic example of an exchange reaction or a displacement where we're making and breaking bonds simultaneously. Redox reactions. So this is a combination of two words. The red stands for reduction, and the ox stands for oxidation. So that's the first thing to see, is we're going to do two things at the same time. Right? We're going to be reducing something through reduction and oxidizing somebody at the same time. So let's think about this a bit more in detail. If you want to think about redox reactions, this is very much like runners passing some kind of a baton in a relay race, except instead of a baton, it's going to be electrons that are going to be passed off back and forth. Now here's the rule, and there's really only one rule and then the opposite of the rule. If you lose electrons, if you hand off the baton, you are said to be oxidized. That's the definition of oxidized. If you are picking up the baton, you are being reduced. And I know that might sound strange at first. You're saying I'm picking up a negatively charged electron, but I'm being reduced. Exactly, you're being reduced in charge. So if you're reduced in charge, we say you have been reduced. Now these two things always happen together. You can't have a relay race and you can't hand the baton to nobody. Somebody's got to be there. So these reactions always happen simultaneously. And usually it's a step-by-step process that we do a series of redox reactions, just like runners in a relay race. And I'll kind of draw like this to get the idea better, that there may not be just, two players in this game. There's a lot of runners in this relay race waiting for it. And they're all passing electrons to each other, but not at the same time. They're doing it one after the other, after the other, after the other. Right? So if you had runner A and runner B and runner C and runner D and runner E and so forth, you could say that at this moment right now, A is passing to B, right? A is passing the baton off. A is passing the electrons off to B. So A is being oxidized because it's losing. And there are ways to remember this. Oil, rig. A very simple trick. O-I-L, oil. Oxidized is losing. It's losing electrons. And rig, R-I-G, reduced is gaining. Reduction is gaining electrons. So at this moment in time, A is being oxidized, but at the very, very same moment, B is being reduced because B has grabbed the baton. Now B is going to run around the track with the baton, and then when he comes back to the starting point, what happens now? Well, now B is being oxidized because he's losing it. And C is being reduced. Right? And C is going to run around the track, and then when he gets back to where he was, C will be oxidized, and D will be reduced, and on and on and on and on and on. Sometimes this is referred to as a redox pathway. And so you'd have many runners passing baton or batons to each other, doing some work, then passing it to someone else and doing some work and passing it to somebody else. These are redox reactions. Okay, of course you can get into hey, I can add water and take away water, and I can make these reactions happen fairly quickly, you know, just in a sink, a kitchen sink. Well, sometimes in a cell or in a life form, an organism, you need it to go faster than just kitchen sink chemistry. So there are ways inside of cells to increase the rate, or the speed if you want to think, of a reaction, so we can get many, many millions and millions of reactions done per seconds, right? So again, we're going to save the details of this for a later discussion. But classically, you hear about how the rate of reaction can be influenced by things like temperature, pH, and pressure. Things we've talked about a little bit before, and things we'll talk about a little bit more in the future. The big player in this story is usually the word enzyme. So an enzyme is really great about influencing the rate of a chemical reaction. They are catalysts. If you want to go further than just saying biological catalyst, you could say they are protein catalysts. And instead of just saying they only speed things up, even though that's what they mostly do, they certainly increase the efficiency of reactions without being changed or destroyed by the reaction. So we can use that enzyme over and over and over again to do lots and lots of work very, very quickly. So the rate of reaction can be influenced by lots of factors, including temperature, pressure, and pH. But biological proteins, called enzymes, protein catalysts, can increase efficiency of a reaction, which usually ends up speeding it up even more, where you can do millions of reactions per second. All right, as usual, thanks for watching. If you want to check out the rest of the series, go ahead and watch the other videos. I'll see you for the next one. Bye-bye.